presentation. That was a good experiment. First time I ever used a video clip with my laptop in here. I learned some new things, like when I turn off my microphone, my earpiece, the recordings automatically stop. You know, you learn something new every day. All right, so where were we? Cell cycle. So we've replicated the DNA. Now we need to go through all the other parts of the cell cycle. It's not just replicating the DNA that we need to do. There's more to it. We need to get organelles replicated. We need to have organelles for each of the daughter cells. And we also need to check to make sure that we did a good job of replicating the organelles and did a good job of replicating the DNA. So there are going to be different phases to this process. So as we look at the cell cycle in lab, we spent a lot of time on mitotic phase. And really, that's where we're going to spend most of our time. But before we get to mitotic phase, we need to spend some time focusing on interphase, which is most of the cell cycle. And during interphase, there's some key things that need to happen. We have G1, S, and then G2. So let's look at G1, the first gap phase. G1 is when that brand new daughter cell is still wet behind the ears. It's brand spanking new. And this cell is going to go through a growth period. It's not at all ready for duplication or replication. So the cell is going to grow larger. It's going to add to its cytoplasm by adding more cytosol and more organelles. It needs to accumulate lots of materials. This is a rapid growth phase for the cell. After it has enough materials accumulated, the cell will leave G1, first gap phase, and enter S phase. When we think of S, think of synthesis. What's being synthesized in S phase? DNA. So during S phase, we synthesize a copy of the DNA. And we just talked about DNA replication. After we replicate the DNA, we go to the second gap phase. And this second gap phase, class, is going to be a proofreading, a double-checking phase. So during the second gap phase, we correct any replication errors. Brand spanking new DNA is slightly different from older DNA. There's something called a methyl tag. So during this G2 phase, we have other enzymes that will scan the molecule of DNA, comparing the brand new side to the older side and fixing any mistakes that were made. It's the proofreading phase. Something else that happens is that we're going to continue to grow the cell. We're going to add more enzymes, more organelles. This is the checkpoint, and we get ready to replicate the cell. Now, if the cell meets all of the chemical checkpoints, the chemical reactions shift towards mitotic phase. Because really, the cell is just a bunch of chemical reactions. And those chemical reaction chains shift towards the mitotic phase, or M phase, where we replicate the nucleus and cleave or replicate the cytoplasm. So during M phase, and this is what we talked about the most during lab, we replicate the nucleus and then pinch off the cytoplasm. Every once in a while, though, we have cells in our body that don't replicate very often. And as you get older, you get more and more of these stagnant cells that don't regenerate themselves. Cells that aren't going to regenerate themselves, instead of going to S phase and duplicating their genomes, will instead will go to G naught or G sub 0. This is an alternative phase where the cell can just be in kind of stasis. And cells can spend days, months, years, decades in this G naught phase. In particular, when you think of neurons of the brain or skeletal muscle fibers, cells that we as adults have a very hard time regenerating. One of the reasons why we have a very hard time regenerating lost brain tissue or lost skeletal muscle fibers is because we have most of our cells in the G naught phase. They aren't actively reproducing. Other tissue types that we can replace easily, like maybe you scratch your skin and then you grow new skin to fill in the wound, those cell types have almost none of their cells in the G naught phase. They're almost all exclusively going to be in the rapidly dividing stages of the cell cycle because we have lots and lots of damage accumulating to those tissue types all the time. So let's move on to mitosis. As we look at mitosis, this is going to be division of the nucleus, and it'll be followed by cytokinesis, the cleavage of the cytoplasm. So as we think of mitosis, to really summarize it in just a nutshell, we're going to take one nucleus and turn it into two nuclei. And we want them to be almost identical to each other. And all the subsequent phases we talk about, all the subsequent steps that we talk about, are going to be stages that we use just to help better organize our thoughts. Keep in mind, even though I'm using absolute terminology in this presentation, it's metaphase, then it's anaphase, then it's telophase. In real life, there's going to be a gradient or a transition. It's not instantly one and then instantly the next. It transitions from one to the next one. So just bear that in mind. There are lots of in-betweenies. Think to lab last week when you look at the whitefish blastula slides. Most of the cells you looked at on those microscope slides were kind of hard to identify because you're like, well, is this in anaphase or is this in metaphase? It's kind of in between. And that's the reality of the situation. So when we think of mitosis, think of division of the nucleus. Why would we divide the nucleus? We divide the nucleus when we need to have daughter cells produced. So after birth, after we're born, we need to generate lots and lots of new tissues. So we have growth of tissues. We're hopefully at the stage where we are not growing that much anymore. Maybe some of the young men in the room will maybe grow another inch or two in their early 20s. But at this point, for the most part, we are going to be replacing cells that die naturally. For example, we replace all the cells that line our stomach every 3 days. All of our red blood cells are going to be replaced every 120 days. This is just a natural part of recycling our bodies. And then if we have some kind of damage occur, we need to repair those damaged tissues by regenerating, hopefully, new cells. As we get older and age, our ability to regenerate new cells declines. And instead of making new cells, we just make fibrous proteins that hold everything together, or scar tissue. Now, during mitosis, we've broken it up into 4 phases: prophase, metaphase, anaphase, and telophase. There are lots of mnemonics out there to help you remember the stages of mitosis. Some of them are G-rated, some of them are more PG-13. Pick your favorite mnemonic that you use to remember these in order and stick with it. For me in particular, it was being pro-mana-T, because I think manatees are cool looking. So I'm pro-manatee. But you do you, whatever works best for you, pick that method. So let's talk about prophase. During prophase, we talked about it in lab, so you can identify it. Let's talk about what's happening, though, the what of prophase. So during prophase, that chromatin is going to be condensing into chromosomes. And we can now see the individual chromosomes. And there's a reason why we do this. It's easier to move around a tightly wound chromosome than it is a loosely wound chunk of chromatin. Think of it like this. Most of your parents, or most of you, probably have some drawer in your house with all the old cords, the RCA cables, the S-video cables, the extension cords. And if you are anything like me, you're like, ugh, I don't have time for this. You just throw it all in the drawer, and it's a hot mess, and you can't get a cable out. You grab one cable, and they all come out instead. But every once in a while, maybe you're visiting a friend's house, because I don't really know anybody in my family that's this organized, but you're visiting a friend's. Actually, my father-in-law is this organized. So I'm visiting my father-in-law's garage, and he has a very nice garage. We call it a garage mahal. And all of his extension cords are tightly wound and bundled. He was a Boy Scout, so he's really good at bundling ropes and cables. And I can reach into his drawer, grab a cord, and pull out only the one extension cord immediately. It's a thing of beauty. When we think of like loosely bound extension cords versus tightly bound extension cords that are nicely packaged, it's kind of the same with chromatin versus chromosomes. We want to take those loose pieces of DNA, wind them up nice and tight, and then move them around after we take the time to wind them up and package them. So that's why we condense the chromatin into chromosomes. As the chromosome forms during prophase, the nuclear envelope disintegrates. The centrioles, which are part of the centrosome, are going to have mitotic fibers, or very long mitotic microtubules, grow out and attach to the centromere. In particular, there's a part of the centromere called the kinetochore that these mitotic spindles attach to. Next, we have metaphase. Let me skip ahead a little bit here. I want to show you a picture. So here we have prophase. Here's the figure from your textbook. So we have duplicated chromosomes. We have mitotic fibers growing from the centrosomes. And in the cell, you can see a bunch of chromosomes in the middle without a nuclear envelope. After prophase, we have metaphase. And in metaphase, those mitotic spindles, before they separate the chromosomes and rip them in half, they're going to take some time to organize the chromosomes. And they make a nice, neat little line of chromosomes right down the middle of the cell. It's kind of like when you're making Christmas cookies. So when you're putting frosting on your sugar cookies, you don't just take a bowl full of sugar cookies, throw it on your countertop, and spray frosting all over the top. Instead, you make a line of sugar cookies, and then you do one, and then you go to the next sugar cookie, and the next sugar cookie. And in the same way, if we take these chromosomes and line them up in the middle of the cell, we can rip them apart and separate them without them tangling each other up. And this is the purpose of metaphase. We line the chromosomes up to minimize the amount of tangles that occur as the microtubules yank them apart. So during metaphase, we have the chromosomes along the equatorial axis of the cell, and the microtubules are getting ready to pull them apart at the centrosome. Anaphase is when the replicated chromosome is physically ripped apart by the microtubules pulling on the kinetochore of the centrosome. Or another way to think of it is think back to your freshman physical science class. Most of you had to take some generic physics class when you were freshmen in high school, and you had kinetic energy and potential energy. Kinetic, or kineto, is a root word that means motion. Kinetic energy is movement of energy of motion. A kinetochore has that root word for motion in it. And as we think of the kinetochore, it is a motor protein which is part of the centrosome, and the kinetochore is what physically is crawling along the microtubule. And this supplies the tugging force that rips apart the sister chromatids. We also have some enzymes involved with this process. These enzymes are going to cleave one side of the chromosome from the other side. They're going to cleave the centrosome right down the mid-sagittal plane. There are many, many proteins and many, many enzymes involved in this process. And this mitosis is still a very active area of research because mutations to mitosis are directly related to the formation of cancerous tumors or cells growing out of control in our body. And if we can better understand this whole process of cells reproducing and how mistakes happen and cells reproduce out of control, we can better make therapeutic medicines. In other words, if we know why it's broken, we have a better idea of how to fix it. So mitosis right now is still pretty interesting. There's still lots of research being done right now. Let's focus on the next phase. Actually, let's hold that thought. Let's look at the anaphase picture. So here's our anaphase picture from your textbook. So we have the chromosome being ripped in half and being pulled to opposite ends of the cell. In our photomicrograph, you can see it in the upper right-hand corner of your screen. After anaphase, we have telophase. Telophase is when those unreplicated chromosomes are being rebound into a new nucleus at opposite ends, or opposite poles of the cell. To make the new nuclear envelope, the rough endoplasmic reticulum is working overtime. And the rough ER is going to shed a lot of its ribosomes and repurpose many of its phospholipids to make a brand spanking new nuclear envelope. Many of the ribosomes that are shed will either go into the new nucleus that's forming and will form the brand new nucleoli, or nucleolus, I should say, in the center. And then other ribosomes will be free floating in the cytoplasm of the cell. After the new nucleus forms, the chromosomes will uncoil to form chromatin. The mitotic spindles disintegrate, and the building blocks are recycled, and each nucleus will have a new nucleoli form. So in our figure, photomicrograph in your textbook, we have that figure which I just circled. I want to emphasize the cleavage furrow. That's the key characteristic I and I believe Ms. Landrich both look for in microscope slides to identify telophase. So we have two new nuclei forming, and at the same time, there's one other thing happening. At the same time telophase is occurring, we're having cytokinesis occur. Telophase is the formation of the two new nuclei during mitosis. Cytokinesis is the pinching off of the cell membrane. So cytokinesis is the final step where we pinch off the one daughter cell and make one parent cell and make two daughter cells. So as we look at cytokinesis, it's the division of the cytoplasm. And it happens when the cytoskeleton that's attached to the cell membrane contracts. We're going to have proteins along the central web, particularly a lot of myosin proteins, one of the motor proteins that we have in our muscular cells. And these myosin proteins will cause the cell membrane in the center of the cell to get narrower and narrower and pinch off, eventually creating the cleavage furrow and then pinching the cell in two. To go back a slide, we have the cleavage furrow shown right here, and then we have the two brand new daughter cells. We do not have time for this video link on mitosis, but it's the same video link that I linked to your lab objective sheets. So either watch it now or watch it from your lab objective sheets. It's just a video showing you the stages of mitosis in CGI. So let's go through our next review question. Cytokinesis overlaps which phase of mitosis? Or another way of phrasing it is the cell membrane pinches during which stage of nuclear replication? Considering I covered this 2 slides ago, the answers are flying in right now, which is good. You have great short-term memory. So we're down to 20 seconds. Is it S phase, telophase, metaphase, prophase, interphase? If you have not yet, please submit your answers now. All right, let's see how we did as a class. You nailed it. Great work. Telophase is the correct answer. Let's move on. So, why do cells divide? Cells are going to divide for all kinds of reasons. We need to heal tissue damage. We have all kinds of potential reasons. But things need to happen before our cells divide. We need to make sure that the DNA has been duplicated. We need to make sure there's enough cytoplasm in the cell so that it has the appropriate surface area to volume ratio to incorporate one of our lab activities. The surface area of the cell and the volume of the cell are 2 very important things that dictate cell replication. They need to have an adequate supply of nutrients. They need to have the right growth factors. If a cell next door dies, hey, there's some fresh real estate to move into. It's kind of like when an empty lot pops up in town, maybe a house burns down, and then all of a sudden there's this big piece of empty dirt. That empty dirt is going to have a new house pop up on it. You know, we are going to take advantage of the available space. Now, these are the things that we need for the cells to divide. Cells will stop dividing when they don't have these criteria met. Generally speaking, physical contact is going to encourage cells to not divide, to halt the replicative cycle. So if there's physical pressure against the plasma membrane, cells are much more likely to enter the G0 phase and just kind of be on the static holding phase. This is one of the reasons why we have such a hard time making new brain cells. Our brain's stuck in a fixed area. It's very hard for our brain to expand outside of that fixed area. Hopefully it doesn't. Something else that causes our cells to stop dividing is lack of nutrients. Improper nutrition will cause the cells not to divide. And I'm not just talking calories. I'm talking micronutrients, the vitamins, the organic molecules, and the minerals, the inorganic components that we need for our cells to function properly. Some other things that help to regulate cell division is a molecular timer. Our cells will sometimes have a pre-programmed stopwatch, for lack of a better analogy, that is going to determine when the cells do and do not enter mitotic phase, or the mitosis and cytokinesis. These molecular timers are referred to as cyclins. They help to regulate the cell cycle. They are cyclins. Just to review nomenclature, proteins oftentimes will have an INS. So if it's a generic protein that's not an enzyme, it typically will have an IN, or an IN, at the end of its name. Actin, myosin, dynin, these are all examples of proteins that are non-enzymes. So, cyclins are the proteins that help to regulate the cell cycle. We have some other proteins, cyclin-dependent kinases, that are going to be turned on and off by the cyclins. So, these enzymes can be activated by the cyclin. It's an enzyme, it ends with an ACE. And as these cyclin-dependent kinases are activated, they are going to move around the cell and help to activate and deactivate all of the other proteins involved with regulating S phase, G1, G2, G0, and there's many, many subcategories. We could spend probably an 8-credit class just talking about this PowerPoint slide right here, but we don't have time on that. This is a black hole of information. So we're just scratching the surface, and then we're going to move on. So as we look at these cyclin-dependent kinases, they regulate DNA replication during S phase. They regulate condensing of chromosomes from chromatin, making the nuclear envelope, making the mitotic spindles. They regulate centromere formation and separation. They do all kinds of different things. Let's move on and talk about how we can control going from G0 to S to G2 to G1. There are checkpoints throughout the cell cycle. These checkpoints are going to be controlled by these cyclin-dependent CDKs. So as we look at these checkpoints, we have, at the very beginning, the G1 checkpoint. So the cell has just divided. It's just gone through telophase and cytokinesis. We have a brand spanking new daughter cell. And that brand spanking new daughter cell will take a moment to assess itself. And it'll kind of assess itself molecularly and go, okay, should I continue to grow, or do I have a bunch of mutations? And should I kill myself? Most of the time, these cells will go, oh, okay, I'm ready, and they'll just continue to the G0 phase. But every once in a while, there are mistakes during mitosis, particularly during metaphase or anaphase. Sometimes those replicated chromosomes aren't ripped apart to make unreplicated chromosomes. And instead, an entire replicated chromosome is pulled to one side, and now there's too much genetic information in one daughter cell, and then the other daughter cell doesn't have enough genetic information. That's an example of when this G1 checkpoint would go, oh, I screwed up. I need to kill myself. And then the cell will go through autolysis because it has incorrect replication or division of the chromosomes. We also could have the G2M checkpoint. This is going to be late in the G2 phase. This G2 checkpoint is going to determine whether or not we're ready. It's the last big checkpoint. It's kind of like when you're getting ready to go on a big road trip. You know, you do some pre-planning, you gather your materials, you get your itineraries, And then right before you sit in the car and start the engine, you kind of check and make sure, okay, is everybody here? Do we forget anybody? Do we forget our sleeping bags? It's the last checkpoint before you move on with cell division. So, my watch says quarter after, and my computer says 14 after. This is a really good natural stopping point. So, Thursday of this week, we will pick up on this slide. What is a gene? And we'll pick up on this slide next class period.